published back in, this is February 2014, and I thought it was a really interesting looking article, so thought you guys might enjoy reading it with me. Let's get started, okay? So this is the new science of the brain. And I think it's on page 28 maybe. I like National Geographic a lot. It's good articles, lots of good, interesting things. Always learn something new, for sure. Yeah, so I'm excited. There, we had to make some adjustments. Sorry about that. And the camera's hanging by like a tightrope, basically, so pardon me. Sorry about that, guys. Okay, this is Secrets of the Brain. New technologies are shedding light on biology's greatest unsolved mystery: how the brain really works. That's interesting. This is a big picture of a very large computer machine. It says it's called the Mind Machine. An engineer wears a helmet of sensors at the Martinos Center for Biomedical Imaging, part of a brain scanner requiring almost as much power as a nuclear submarine. What does it say? Antennas pick up signals produced when the scanner's magnetic field excites water molecules in the brain. Computers convert this data into brain maps like the ones you'll see here. Wow. Do you see? His eyes are right here, so then this is all in his head. That's wild. That's a lot of computers. Oh, and here's a big... This is a big picture of the brain. Really interesting. Got 1, 2, 3, 4, 5, 6, 7, 8, 9. So number 1 is the frontal cortex. Number 2 is the motor cortex. Number 3 is the parietal lobe. Number 4 is the corpus callosum. Number 5 is the thalamus. Number 6 is the occipital lobe. Number 7 is the temporal lobe. Number 8 is the brainstem. And number 9 is the cerebellum. Wow. And this picture, it says the color of thought. The brain's many regions are connected by some 100,000 miles of fibers called white matter, enough to circle the Earth 4 times. Images like this taken at the Martinos Center reveal for the first time the specific pathways underlying cognitive functions. The pink and orange bundles, for example, transmit signals created for language or critical for So that's kind of interesting, is how you know where the language is centered in the brain. So if someone has a stroke or a brain injury, that part of the brain is damaged, then they may lose their understanding or ability to process language. There's an old picture of the brain to drawing. Pretty accurate though, I think. Looks like this was drawn in the 1800s, it says. This article's by Carl Zimmer with photographs by Robert Clark. Van Whedon strokes his half-gray beard, and he leans towards his computer screen, scrolling through a cascade of files. We are sitting in a windowless library, surrounded by speckled boxes of old letters, curling issues of scientific journals, and an old slide projector that no one has gotten around to thawing out. It'll take me a moment to locate your brain, he says. On a hard drive, Whedon has stored hundreds of brains, exquisitely detailed 3D images from monkeys, rats, and humans, including me. Whedon has offered to take me on a journey through my own head. We'll hit all of the tourist spots, he promises, smiling. This is my second trip to the Martinos Center for Biomedical Engineering, Imaging, located in a former ship rope factory on Boston Harbor. The first time, a few weeks ago, I offered myself as a neuroscientific guinea pig to Whedon and his colleagues. In a scanning room, I lay down on a slab, the back of my head resting in an open plastic box. A radiologist lowered a white plastic helmet over my face. I looked at him through 2 eye holes as he screwed the helmet tight so that the 96 miniature antennas it contained would be close enough to my brain to pick up the radio waves it was about to emit. As the slab glided into the cylindrical maw of the scanner, I thought of the man in the iron mask. The magnets that now surrounded me began to rumble and beep. For an hour I lay still, eyes closed, and tried to keep myself calm with my own thoughts. It wasn't easy. To squeeze as much resolution as possible out of the scanner, Whedon and his colleagues had designed the device with barely enough room for a person of my build to fit inside. To tamp down the panic, I breathed smoothly and transported myself to places in my memory, at one point recalling how I had once walked my 9-year-old daughter to school through piles of blizzard snow. As I lay there, I reflected on the fact that all of these thoughts and emotions were the creation of a 3-pound loaf of flesh that was under scrutiny. My fear, carried by electrical impulses converging in an almond-shaped chunk of tissue in my brain called the amygdala, and the calming response to it marshaled in regions of my frontal cortex. My memory of my walk with my daughter was coordinated by a seahorse-shaped fold of neurons called the hippocampus, which reactivated a vast web of links throughout my brain that had first fired when I had clambered over the snowbanks and formed those memories. I was submitting to this procedure as part of my cross-country reporting to chronicle one of the great scientific revolutions of our times, the stunning advances in the understanding of the workings of the human brain. Some neuroscientists are zooming in on the fine structure of the individual nerve cells, or neurons. Others are charting the biochemistry of the brain, surveying our billions of neurons produce and employ thousands of different kinds of proteins. Still others, Whedon among them, are creating an unprecedented, detailed representations of the brain's wiring, the network of some 100,000 miles of nerve fibers called white matter that connects the various components of the mind, giving rise to everything we think, feel, and perceive. The U.S. government is throwing its weight behind this research through the brain research through advancing innovative neurotechnologies BRAIN initiative. In an announcement last spring, President Barack Obama had said that the large-scale project aimed to speed up mapping of our neural circuitry, giving scientists the tools they need to get a dynamic picture of the brain in action. Hello, Shadow. Would you like to read? Okay. You can read with me. As they see the brain in action, neuroscientists can see its flaws. They are starting to identify differences in the structure of ordinary brains and the brains of people with disorders such as schizophrenia, autism, and Alzheimer's disease. As they map the brain in greater detail, they may learn how to diagnose disorders by their effect on the anatomy and perhaps even understand how those disorders arise. On my return trip to his lab, Whedon finally locates the image from my session in the scanner. My brain appears on his screen. His technique, called diffusion spectrum imaging, translates radio signals given off by the white matter into high-resolution atlas of that neurological internet. His scanner maps bundles of nerve fibers that form hundreds of thousands of pathways, carrying information from one part of my brain to another. Whedon paints each path a rainbow of colors so that my brain appears as an explosion of colorful fur like a psychedelic Persian cat. Whedon focuses in on particular pathways, showing me some of the circuitry important to language and other kinds of thought. Then he pares away most of the pathways in my brain so that I can more easily see how they're organized. As he increases the magnification, something astonishing takes place before me. In spite of the dizzying complexity of the circuits, they all intersect at right angles, like the lines on a sheet of graph paper. It's all grids, says Whedon. When Whedon first unveiled the grid structure of the brain in 2012, some scientists were skeptical, wondering if he'd uncovered only part of a much more tangled anatomy. But Whedon is more convinced than ever that the pattern is meaningful. Whenever he looks in the brains of humans, monkeys, rats, he finds the grid. He notes that the earliest nervous systems in Cambrian worms were the simple grids, just a pair of nerve cords running from head to tail, with rung-like links between them. In our own lineage, the nerves at the head end exploded into billions but still retained that grid-like structure. It's possible that our thoughts run like streetcars along these white matter tracks as signals travel from one region of the brain to another. There's zero chance that there are not principles lurking in this, says Whedon, peering intently at the image of my brain. We're just not yet in a position to see the simplicity. Scientists are learning so much about the brain now that it's easy to forget that for much of history we had no idea at all how it worked or even what it was. In the ancient world, physicians believed that the brain was made of phlegm. Aristotle looked on it as a refrigerator, cooling off the fiery heart. From his time through the Renaissance, anatomists declared with great authority that our perceptions, emotions, reasoning, and actions were all the result of animal spirits, mysterious, unknowable vapors that swirled through the cavities in our head and traveled through our bodies. The scientific revolution in the 17th century began to change that. The British physician Thomas Willis recognized that the custard-like tissue of the brain was where our mental world existed. To understand how it worked, he dissected the brains of sheep, dogs, and expired patients, producing the first accurate maps of the organ. It would take another century for researchers to grasp that the brain is an electric organ. Instead of animal spirits, voltage spikes travel through it and out into the body's nervous system. Still, even in the 19th century, scientists knew little about the paths that those spikes followed. The Italian physician Camillo Golgi argued that the brain was a seamless, connected web. Building on Golgi's research, the Spanish scientist Santiago Ramon y Cajal tested new ways of staining individual neurons to trace their tangled branches. Cajal recognized what Golgi did not, that each neuron is a distinct cell, separate from every other one. A neuron sends signals down its tendrils known as axons. A tiny gap separates the ends of the axons from the receiving ends of the neurons, called dendrites. Scientists would later discover that axons dump a cocktail of chemicals into the gap to trigger a signal in the neighboring neuron. Jeff Lichtman, a neuroscientist, is the current Ramon y Cajal Professor of Arts and Sciences at Harvard, carrying Cajal's project into the 21st century. Instead of making pen and ink drawings of neurons stained by hand, he and his colleagues are creating extremely detailed three-dimensional images of neurons, revealing every bump and stalk branching from them. By burrowing down to the fine structure of individual nerve cells, they may finally get answers to some of the most basic questions about the nature of the brain. Each neuron has on average 10,000 synapses. Is there some order to their connections to other neurons? Or are they random? Do they prefer linking to one type of neuron over others? To produce the images, Lichtman and his colleagues load pieces of preserved mouse brain into a neuroanatomical version of a deli meat slicer, which pairs off layers of tissue, each less than 1,000th the thickness of a strand of human hair. The scientists use an electron microscope to take a picture of each cross-section, then use a computer to order them into a stack. Slowly, a three-dimensional image takes shape, and one that the scientists can explore as if they were in a submarine traveling through an underwater kelp forest. Everything is revealed, says Lichtman. The only problem is the sheer enormity of everything. So far, the largest volume of a mouse's brain that Lichtman and his colleagues have managed to recreate is about the size of a grain of salt. Its data alone total 100 terabytes, and the amount of data that is the amount of data in about 25,000 high-definition movies. Once the scientists have gathered this information, the really hard work begins, looking for the rules that organize the brain's seeming chaos. Recently, Lichtman's postdoctoral researcher Narayanan Kasturi set out to analyze every detail in a cylinder of mouse brain tissue measuring just 1,000 cubic microns, a volume 1/100,000th the size of a grain of salt. He selected a region surrounding the short segment of a single axon, seeking to identify every neuron that passed through it. That minuscule patch of brain turned out to be like a barrel of seething snakes. Kasturi found 1,000 axons and about 80 dendrites, each making about 600 connections with other neurons inside the cylinder. It's a wake-up call to how much more complicated brains are than the way we think about them, says Lichtman. That's really amazing. Where is the end of all the ways? I don't know where it stops. Because that's not the end. That's page 43. This is like a big picture. So where's the end of there's page 51. Must be another picture. 55 and 56. So we have oh my gosh, 8 more pages to go. So I think we'll have to stop for now, guys. I know, don't whine. But because this is long and it's getting late, you guys need to go to bed. Get your rest, okay? But tomorrow we can look over the second half and then look at some of these pictures. There's a whole page on the bionic brain and how the different body parts are controlled by the body and the spinal cord. This is very, very fascinating and incredible stuff. It's incredible. Imagine being a brain surgeon. I mean, I can't even fathom it. It's incredible. Incredible. So, it is truly amazing. Thought, feeling, sense, action all derive from unimaginably complex interactions among billions of nerve cells. A section of mouse brain, no larger than a grain of salt, serves as a window into this hidden world. That's pretty amazing. I hope you guys liked it. I thought it was really good. Super interesting. I'm just fascinated. And I think that our friend because she's really interested in psychology and the brain and how it works and all kinds of things. So she even went to school for it. And so I hope that she especially likes it. Wish her well and look forward to finishing this with you guys again on the next video, okay? Great. Thank you for your understanding and being so patient. And then there's also another article I want to read. This one here. It's called No Place Like Home, I think. And there's an article about this town just north of Minnesota, Minneapolis. Just kind of looked good. And I once lived there a long time ago. Oh, see this little girl. Oh my gosh. So sweet. Holding that bird is amazing. So, this is incredible. It's incredible stuff. So, thank you guys for listening, for being so quiet. You did such a nice job. You paid attention. Thank you. I appreciate you very much. Okay. Thank you guys, and thank you too for being here, for listening with me. I hope you enjoyed this article and the reading. I hope you found it restful and peaceful and relaxing. And I'll very much look forward to reading the rest of this article for you and some others perhaps coming up soon. So, in the meantime, please stay warm, sleep well, rest well, and thank you so much for watching. Please like,